Let's talk about our other type of passive transport, facilitated diffusion. So some molecules, those that are small, those that are non-polar, and those that are uncharged, can move very easily across a plasma membrane. That's not to say that polar molecules and other small polar molecules in particular can't move across the membrane. In fact, water can, small charged molecules can, but they move much more quickly if they are moved by facilitated diffusion. Small molecules like carbon dioxide and oxygen are nonpolar. Those gases can move easily across a membrane. Water molecules, although small, move slowly because of their charge, because of their polar properties. They're very easily blocked by that hydrophobic center of the plasma membrane. In facilitated diffusion, we see the use of a channel protein that allows for the movement and transport of molecules across a membrane. Because polar molecules and ions are essentially blocked by that lipid bilayer, the use of proteins allows for a much quicker and more specific movement of those molecules. But keep in mind that they're still moving by diffusion. And we're moving by passive diffusion, which means, diffusion is always passive, which means there's no energy required for it. Diffusion is driven by movement via the concentration gradient. In facilitated diffusion, the transporters are usually specific. They don't have to be, but they are specific to a nonpolar hydrophilic molecule. Our first type are channel proteins. So these are basically corridors or tunnels through the plasma membrane. They are not usually as specific as our other types of transport proteins, and they can't block other nonpolar molecules from coming through, but they are generally slightly specific. They might allow a certain size molecule or something like that. But the inside lining of that channel protein is also going to be hydrophilic. Remember that membrane is hydrophobic, and so that protein provides a channel through which those hydrophilic membranes can move unimpeded. These molecules, again, are going to move from area of high concentration. We see a higher concentration of the orange triangles there on the top side of that plasma membrane, or the outside of that plasma membrane, and then a lower concentration inside. So that is what's driving this movement. Channel proteins are often ion channels, which are specific and allow only certain types of ions to pass through. An example of this are gated channels. These are found in nerve cells, and they're important in sending messages along those nerve cells. In a gated channel, we have a slightly modified version of a channel protein where the ion channel opens in response to some sort of stimulus. And when that channel opens, it allows the ion to move through via diffusion. Our other type of channel protein, our other type of transmembrane protein for facilitated diffusion is the carrier protein. In a carrier protein, we see much more specificity. These proteins are going to bind a specific molecule. Here we have that round blue molecule. And when they bind the molecule on one side, it causes a shape change that allows it to open up and move the molecule out the other side. I like to think of these as those sort of revolving doorways that you find at a hotel. It sort of impedes the flow. Molecules of a certain size can only fit through. They can't carry their luggage through. And as you enter, it moves and you get spit out the other side. That's a carrier protein. Carrier proteins can actually be active diffusion as well, and we'll talk about those in just a minute. In active transport, we see the specificity of our carrier proteins But in active transport, we require energy to move the solute across a membrane. And the reason that we require energy is that in active transport, we're moving something against its concentration gradient. So ions and molecules, water, all of these things want to move down their concentration gradient from areas of high concentration to areas of low concentration. But in active transport, the cell is trying to move something against its concentration gradient. This might be a waste product, or it might be trying to accumulate something in the cell. And so energy is required to do this. Because they're moving specific molecules, these carrier proteins involved in active transport are specific. And so we see the binding and then a shape change in the movement of that molecule. So in this example, we see that the cell is trying to move the square molecules in and the round molecules out. So on the top side, it would open up, bind the square molecule, and then change shape, release the molecule out the other side. After that happens, it can bind the circular molecule, change shape, and move it to outside of the cell. This is really important because it allows the cell to regulate concentrations of ions and to maintain them at a different concentration than outside the cell. If this were passive transport, molecules moving by diffusion, you would eventually reach equilibrium. But active transport allows the cell to maintain something besides equilibrium. The energy used in this example is ATP. We're going to talk about ATP a lot in the coming chapters. ATP is basically our energy currency in the cell. It's the molecule that is used to power the majority of reactions. So one example of active transport is the sodium-potassium pump. In the sodium-potassium pump, we build up ions on either side of the cell membrane in order to produce a membrane potential. We're not going to go into the details too much, but a membrane potential is what allows the signal in a neuron to be passed between neurons. So some sort of stimulus, external stimulus, alerts a neuron, and it causes this flow and change in ions across the membrane. And it's that flow and change that actually propagate that signal down your nerve to the next nerve. We don't talk about membrane potentials in this class. That's a topic for 102. It is found later in your book if you are interested in how these work in a little bit more detail. But the purpose of this pump is to build up a concentration of potassium inside the cell and sodium outside the cell. And then when the cell is stimulated, those ions will flow through because they have a charge, because they have an energy potential, and that's what moves the signal. So starting at box 1 on the top center, we're trying to move sodium outside of the cell, so moving it from the inside to the outside. So that channel protein is going to bind that sodium. A shape change is going to occur when energy is added to the system, so ATP is added to the system. That shape change allows the sodium to exit outside of the cell. And then a second shape change occurs that allows potassium to bind and move from the outside in. In this case, we see more energy being used. That's the indication of the phosphate to the PI. That's the using up of the energy. And then the potassium is going to be deposited inside the cell. Again, this allows the cell to maintain very specific concentration gradients that are different from the outside of the cell and limit the ability of the cell to form an equilibrium. These ion pumps are really important in forming what we call a membrane potential. This is simply a voltage or a charge across a membrane that's created by those charged ions, by those anions and cations. In the sodium-potassium pump, they're both positively charged, and so it's a difference in positive charges across the membrane. But this charge buildup is often done with ion pumps moving hydrogen ions, hydrogen protons across the cell. Remember that those hydrogen ions are positively charged, and so if you move hydrogen ions, you're going to create a buildup of positive charge on one side of a membrane. These ion pumps are working by active transport, so energy is used. And we are going to see these extensively when we look at the processes of cellular respiration and photosynthesis. Both of those processes use this hydrogen ion gradient to power the production of ATP, and that's really important in the cell. Another specialized type of active transport is called cotransport. In cotransport, you use the buildup of an ion gradient to move a secondary molecule. So this picture seems backwards to me, but let's start at the bottom at that proton pump. This is the same proton pump from the previous image. This is a hydrogen ion pump, so it is going to produce a hydrogen ion gradient. High concentrations of hydrogen outside, low concentrations of hydrogen inside. Remember, this requires ATP. On the outside of the cell now, we have a charge, a higher concentration, higher charge of the more positive charge, I should say, created by those hydrogen ions. And remember, when we have molecules in a concentration gradient, they're going to want to diffuse. They're going to want to move back down their concentration gradient to reach equilibrium. So the cell can use this idea in the concept of cotransport. So in this situation, those hydrogen ions are now going to move from high concentration here through a cotransporter. And when that happens, the power of that diffusion, the power of the movement of those ions, is going to move sucrose as well. It's going to allow sucrose to enter the cell, being driven by the movement of those hydrogen ions. Not all of the transport into and out of the cell is mediated by proteins. Smaller molecules and molecules that can move sort of one at a time are going to be moved by either passive or active transport through those transmembrane proteins, those integral proteins. We can also have sort of a bulk import and export of molecules. So in this way, it allows cells to sort of grab or gulp things around them and bring them into the cell. They can also release molecules or products in the same way. So in chapter 4, we talked about vesicles. Vesicles are a little sort of pocket or package of phospholipid membranes that move products throughout the cell. In the process of exocytosis, those membranes are going to move, in this example it's from the Golgi apparatus, to the plasma membrane, and they're going to fuse with that plasma membrane. When they do, they sort of empty or spit out their contents into the extracellular fluid. This is how waste is removed from the cell. This is also how cellular products that are produced in the Golgi apparatus are released from the cell. So sometimes the cell is producing things that need to be used outside of that cell in the bigger organism. And this is one way that those products are removed from the cell. The opposite process can take place when cells need to engulf larger objects. So sometimes a cell will sort of reach out and grab a food particle, something that it wants to consume. And so the process of endocytosis is where a cell sort of encloses something and then produces a vesicle that's now on the inside. In endocytosis, we often call this cell eating. There's a specific type of endocytosis called phagocytosis, and that's where the cell brings in this food particle. Endocytosis is not necessarily specific. It might be engulfing a specific sort of prey item, if you will, but it's not necessarily bringing in only that particle. It can bring in other things as well. Our picture there shows an amoeba eating a green algae cell, and it's doing that by sort of wrapping its arm-like structures around the cell, and then it's going to engulf it and produce a food vacuole. So remember, vacuoles are little storage pockets. They're slightly different from vesicles. Vesicles are generally in movement and temporary, but vacuoles are produced for storage. So endocytosis is producing a little storage packet or pocket within the cell. There are two other types of endocytosis. One is penocytosis. This is equivalent to cell drinking. In phagocytosis, a food particle is brought in. In penocytosis, the fluid surrounding the cell is brought in. So this is not necessarily specific. Basically, the vesicle forms around whatever is surrounding the cell, and so different particles and molecules can be brought in that way, in addition to coming in through transmembrane proteins. Finally, we have receptor-mediated endocytosis. In receptor-mediated cytosis, it's sort of specific penocytosis. There are receptors on the outside of a cell that will bind a very specific molecule. And when those molecules are bound, a vesicle will form around it and bring in those substances. So it allows the cells to bring in large quantities of something specific, whereas penocytosis is nonspecific. An example of this is cholesterol in the diet. So cholesterol is important in the plasma membrane. It's important to be modified and turned into a hormone. And so the cell actually needs to bring in these molecules. It needs to specifically bring these in to be used within the cell. Cholesterol is brought in by receptor-mediated endocytosis. And in this process, it has specific receptors for LDL cholesterol. So this is our good cholesterol. And in a normal cell, the cell has these receptors on the outside. The cholesterol binds and eventually brings it in. In some individuals, this sort of high cholesterol that they get is actually a genetic disorder called familial hypocholesterolemia. Say that 10 times fast. In this disease, the cell doesn't have the receptors or doesn't have enough receptors to bring in the cholesterol that it needs. So you can see the normal cell there. Mild disease, there are limited receptors, and then severe disease, there are no receptors. When those LDLs are not brought into cells, it accumulates in the bloodstream, and this can lead to heart damage and stroke because those lipid molecules actually can form deposits on the walls of blood cells, and this sort of narrows the vessels and impedes blood flow.